Hi students, I hope I'm audible, visible. I hope I'm audible, visible. I hope I'm audible, visible. Hello everyone. Hello. Hello. I hope I'm audible and visible to everybody. Guys, am I? Okay, so if I'm perfectly audible and visible, do let me know in the chat section. Yes, everyone, do let me know in the chat section. Am I perfectly audible and visible? Because the chapter that we are going to discuss today is a very important chapter. So it is the revision session. And today, right, we are going to complete acid soluble fractions. Like we will start from the basics, right? So we will talk about acid soluble, acid insoluble fractions. That's what we are going to cover today. Yes, acid soluble, insoluble fractions that we get from chemical analysis. That's what we are going to discuss today. We'll be talking about primary and secondary metabolite, right? So it is going to be a quick revision session so that, right, you understand what exactly is there. And then we are also going to talk about the carbohydrates. So from basics to the, right, from basics to the advanced level, we are going to cover it. So this is the topic for today. So you tell me, am I perfectly audible, visible? Yes, am I perfectly audible, visible? So I would like to see the responses here, and do not forget to hit the like button as well. So make sure you hit the like button as well, right? Mark your attendance by hitting the like button and be focused. Open your NCERT. So here in this particular chapter, that is of biomolecule, beta, in your NCERT, especially of class 11th NCERT biology, right, the things are not given in that detail, but we need to understand, right, we need to understand the basics if we really want to decode that NCERT. So this particular chapter, which is biomolecules, right, what is the name of the chapter? The name of the chapter is biomolecule. And let me tell you, beta, this chapter is also there in class 12th chemistry. So today itself in the batch, I was showing this to my students as well, that in class 12th biomolecules chemistry, you people understand monosaccharide, oligosaccharide, polysaccharide. You need to learn that. Okay, so it's better to have good command over this particular chapter because it is literally very, very, very important. Okay, so excited for the chapter? Excited to start the basics here? Yes, everyone. So I would like to see the responses here in the chat section. So quickly tell me, excited to understand this wonderful chapter? And biochemistry is very important for all of you, right? Biochemistry is literally very important for all of you, right? Not just in class 11th and 12th, but in the MBBS also. So if you are clear that definitely I will take admission in the in my dream medical college, then definitely you need to, or you should prepare this particular chapter, right? You should prepare this particular chapter. Okay. So even in your MBBS, you are going to learn biochemistry, right, about the amino acids, so many things and processes. Then only you'll be able to understand how this human body is working, right? What is it made up of? Okay. So things are related. So it is literally very important. Okay. Okay, so let's start the chapter. So the word here is biomolecules. So you tell me, people, what do you understand with this particular word? Yes, everyone, what do you understand with this particular word? Quick. What do you understand with this particular word? That is biomolecule. I'll start from the basic. I'll start from the scratch. And I am here going to start from evolution. See, in our day-to-day life, you all notice something. Even if I talk about the mobile phones, right? Initially, no mobile phones, then landlines, the changes occurred in that, then mobile phone, their form, their working, it changed a lot. Keypad phone was there. Now we have the smartphone. It is not just used for the calling, video calling, searching, buying, so many things, right? This is what we are doing. That is how we are using a phone, right? So we have witnessed that, the changes in the phone. So don't you think that type of, if we can do changes in the things which we have made. So don't you think that nature can also do the same? Of course, nature can also do the same, right? In nature also we can see the same things, don't you think so? Definitely we can do the same thing in the nature. If we humans can modify the things, if we humans can change the things, so obviously beta nature, who's the creator, so definitely they can also do the changes. But ma'am, we were talking about the biomolecules. Why are we talking about evolution? Why are we talking about these changes? Let me tell you. See students, if I talk about the earth, right, initially nothing was there. Initially there was no life. We talk about a theory, that is Big Bang theory, right? And we discussed that there was a kind of massive explosion which we cannot measure in physical terms, and after that the cooling occurred, like a gaseous cloud was there, some sort of explosion was there, and with time the material scattered, and what happened is that the cooling occurred with time, the temperature came down, many changes occurred. Initially earth was having no atmosphere, then slowly reducing atmosphere formed, then the oxidizing atmosphere formed. So with time, with a lot of time, things changed. So initially what happened was, right, that what was there on the planet Earth, all these atoms and all, right, all these chemicals were there. Basically students, the life, it started in the water, right? It started in the water, and not immediately after that Big Bang theory, after that explosion. No, it took time, right? It took time for the temperature to come down. It took time so that atmosphere formed with time. So that is how things changed with time. So life evolved in water. Life started in life started in the water. So what happened was condition changed with time. So what happened was that atoms, they were there. That atoms together, they started making molecules, bacha. That molecules, they started making elements. That elements, they started making compounds. And when we talk about the compounds, we have, right, when we talk about the compounds, we have inorganic as well as organic compounds. That is what we have. Inorganic as well as organic compound. And even here in organic compound also, we have simple organic compound, we have complex organic compounds. So that's how things change with time. So yes, we talk about the atom, we know that it is the smallest unit of matter. But when you talk about something as living, you talk about the cell. But ultimately that cell, that life has also started when these chemicals, they form. Our DNA, when it formed, DNA, because it stores the information. So if you talk about a living being, no doubt we start that life starts from a cell. A single cell is the basic unit of life, right? A cell is something which we are calling as life, but ultimately that is also made up of these things. So what happened initially in the water? What happened? Atoms, they combined, right? Because at that time temperature was high, right? Volcanic eruptions were going on, lightning was there. So all that things they provided, right? They work like a kind of catalyst. They helped in these processes. So atoms, they started making molecules, then elements, then compounds, inorganic compounds, organic compounds they formed, and simple organic compound, complex organic compounds they formed, like your I would say your nitrogenous bases, right? Nucleotides, these monosaccharides, these amino acids, all of them they started forming. And meanwhile, let's suppose if you are having monosaccharides, with time, definitely they formed polysaccharide, right? Amino acid, they made proteins. These nitrogenous bases, these sugars, all of them together, right? They started making the nucleotides, and then came your DNA and RNA. So DNA, which is having all the which is having all the genetic information, it is what? It is also, DNA is also what? It's a chemical. It is something which can store the information. So with time, what happened is some processes, they, with time, they evolved, right? These chemicals, they started storing the information. Then some other chemicals formed that started transferring the information. I'm talking about the RNA here, right? Some chemicals formed like proteins, which behave like enzyme, which speed up the chemical reaction. So all of them formed, and they resulted in developing in such a dynamic, I would say, a beautiful system which we call as cell. Are you getting my point? So atoms, molecules, elements, compound, inorganic, organic, all that things together, they made something which we call as protoplasm. And this protoplasm is what? I always mention this. It is the first living substance, right? It is the first living substance, or you can also call it as living content of a cell. You can also call it as living content of a cell. So this protoplasm is the physical basis of life. So whenever I say that something is living, I talk about this protoplasm. So this protoplasm is ultimately having these chemicals, having these organic, inorganic things, right? And that's how we got this life. Right, and that's how we got cell, we got tissue, we got organ system and the organism. So we cannot say that living things, they are not made up of these molecules. Yes, they are made up of, and the molecules that make the living body, we call them as biomolecules. Is that clear? We call them as biomolecules. Is that clear? Is that clear? So here in this particular class, we will revise, we will learn the things from the scratch. Moreover, we will also highlight the NCERT. So if we talk about the NCERT, we have some simple things here, that there is a wide diversity in living organism in our biosphere, right? And they all living organisms, they are made up of same chemicals, that is elements and compounds. This is the question. So how can we prove that question? So now comes the chemical analysis which we have to study. But overall, is it clear, Maitri? Yes, beta. Is it clear? So ultimately, what is there? So we are talking that all the living beings, they are made up of certain molecules, and we are calling that as what? We are calling that as, yes beta, we are calling them as biomolecules. Is that clear? We are calling them as biomolecule. So the first thing now that we have to understand here is that how to analyze the chemical composition. So that's the first thing that we are going to discuss. How to analyze the chemical composition, right? That's what we need to understand here. So how to analyze the chemical composition. So what are we going to do? We are going to perform a very simple experiment here. What's that experiment? Now, the title itself is saying how to analyze chemical composition, because we have to see whether living things, non-living things, what kind of chemicals are making them, do they have same type of chemical composition, or even if same type of chemicals are there, what about their quantity? So there were so many things, right? So scientists, you know that they are curious, they try to figure that out. So that's what we are going to learn here. Is that clear? So this chemical analysis is very important. So do pay attention, okay? Do pay attention, okay? Done. So what are we going to do? First of all, you will take number one, a living tissue. You have to take living tissue, living material. It can be anything, students. It can be anything. Anything as in, it can be plant material, it can be animal material, it can be microbial paste, anything which is living, we have to take that, right, bicha? Right? So it can be a vegetable, piece of liver, anything you can take. It should be a living tissue, right? Vegetable, or liver, anything can be there. So you have to, right, take the living tissue. Okay. Now what we have to do? We will take the living tissue. So take a living tissue, number one, then grind this living tissue with a chemical. Grind this living tissue with a chemical. So what is the name of that chemical? The name of that chemical is trichloroacetic acid. What is the name of that chemical, students? The name of that chemical is trichloroacetic acid. So it's a very simple way to remember this. So acetic acid formula is CH3COOH. Am I right? CH3COOH. What is that? That is the acetic acid. Now trichloro, tri means three carbons. Three trichloro, three chlorines. So CC3Cl3COH. So that is your trichloroacetic acid. So obviously from your acetic acid you are getting trichloroacetic acid. So this is the formula. So what are we saying here? That grind this living tissue with a chemical. What is the name of that chemical? Trichloroacetic acid. So you are going to grind it. You will take a mortar or pestle, right? In that the living tissue will be there. Let me show you that. So let's suppose this, right? So you have the living tissue. You have the living tissue here. Now you are putting the chemical here. Now you are going to grind it in this mortar and pestle. Now after that grinding, what will you do? Anyone in the class who know this? After this grinding, what will you do? You will try to separate that, because when you are grinding, you will make a kind of slurry, right? You will make a kind of slurry. Now you will try to filter that out. What will you do, Sanya? You will try to filter that out. So when you will grind it, you will obtain a thick slurry. So grind this living tissue with a chemical. Grind this living tissue in a chemical. Sorry, with a chemical in the mortar and pestle. Mortar and pestle. Then what will you get, beta? A thick slurry, a kind of, paste. A thick slurry is obtained. That is what you guys are going to do. Now, after that, what will you do? You will try to filter that out, right? That slurry will be there. That slurry will be there. Now we have to strain this slurry. We can use cheesecloth. We can use cotton cloth, anything. So strain the slurry with cheesecloth or cotton cloth, anything you can use. Okay. So we have to strain it. So after that, what will you get? We will get two fractions. What are we going to get, beta? We are going to get two fractions here. So let's understand which fractions are we going to get, right? So you're trying to strain something, right? You are using a cloth here. Imagine, just say you are using a cloth here, you are trying to pour something here. So the thing that will pass down, can I call it as filtrate? Can I call it as filtrate? And the substance which will be left here in this cheesecloth, can I call it as retentate? Yes, definitely. This is the remaining part, and this is what we call as filtrate. So after straining the slurry with the cheesecloth, what you people will get? You will get two fractions here. You will get two fractions here. So one thing is, students, one thing here is the filtrate. One thing is what? Everybody please pay attention. Filtrate. Another thing is will be retentate. So filtrate is there or retentate is there. Now ma'am, what's the difference? Why we got two fractions here? Why is it happening like that? Let me tell you. So now you are trying to strain it. As I said, this is going to be the filtrate, and here you have retentate. So do remember one trick, beta. Do remember one trick. What is that trick? The trick is fast. What is that trick? The trick is fast. What does it mean? It means F stands for filtrate. AS means acid soluble. And from T, you can remember the chemical, that is trichloroacetic acid. That's what you people can remember. So what's the trick that we have here? The trick here is filtrate. The trick here is fast. So what does it mean? Filtrate is acid soluble, and which acid we have used? Trichloroacetic acid. Now this filtrate, because I told you two fractions are there. So this is acid soluble and it is acid insoluble. It is acid soluble and it is acid insoluble fraction. Two fractions are there. How many fractions are there? Two fractions are there. Acid soluble or acid insoluble. So in that filtrate, you are having all the things which are soluble in the acid. In the retentate, you are having all the things which are not soluble in the acid. But why is it so? Why is it so? Now this is what we need to understand. Is that clear? Is that clear? Yes or no? Is that clear? So in the case of acid soluble pool, acid soluble fraction, any word you can use, acid soluble pool or acid soluble fraction. Now here what do we have? Obviously organic compounds, but just organic compounds? No, inorganic compounds are also there. So one by one we will study that. So filtrate, as I said, everybody pay attention, acid soluble pool. So it includes biomolecules having molecular weight less than 1000 Daltons. Less than 1000 Daltons. Are you getting my point? So it includes biomolecules having molecular weight less than 1000 Dalton, and these biomolecules are biomacromolecules. So in the acid soluble pool, you are going to get the biomolecules whose molecular weight is less, and it is lesser than 1000 Dalton. It is lesser than 1000 Dalton, right? So this is basically the unit here. So the molecules having molecular weight less than 1000 Dalton, they are present in the acid soluble pool. So for an example, if I have to give you the example, so here you people will see monosaccharides, right? And it is not just the only things which are present here. There are many. Just say you will find out the amino acids there, right, students? Even the nucleotides here. So they are present here, etc. Right? So their molecular weight is less. Okay, their molecular weight is less. Am I clear? Their molecular weight is less. Is that clear? Right? So here in this retentate, what do you have? Beta, in this retentate, it includes organic compounds. It includes organic compounds having molecular weight. Yes, students, having molecular weight above 10,000 Dalton. Above 10,000 Dalton, not 1,000 Dalton. It is 10,000 Daltons. They are basically the polymers. Yes, please. So it includes organic compounds having molecular weight above 10,000 Daltons. So they are called biomacromolecules. What do we call them? They are called biomacromolecules. Am I clear here? Yes, students. So example include is you will be having polysaccharides here. You will be having proteins here, nucleic acid here, etc. But there is one more thing that you need to remember. So in the acid soluble pool, what do you have? This will have, the organic compounds having molecular weight less than, right, 1000 Dalton. So this will be monosaccharide, amino acid, sugars, right? That is exactly the monosaccharide, nucleotides, etc. But here you have the organic compound having molecular weight above 10,000 Dalton. Now, one more thing that you need to write is lipids, which are not strictly biomacromolecules. This is a very important point, so everybody pay attention. So lipids, which are not strictly biomacromolecules, are biomacromolecules and having molecular weight less than 800 Daltons. Imagine their molecular weight is also less, are also present in it. Imagine. So that's what you need to remember. We'll talk about the reason as well. So in the acid insoluble pool, that is in the retentate, lipids are also present, right, students? Lipids are also present, which are not strictly biomacromolecules. They are not having the high molecular weight. Rather, their molecular weight is even less than 800 Dalton, but still they are present here. Anyone in the class who can revert, who can tell me why is it so? Why is it so? So two fractions are there after the chemical analysis. In one fraction you have acid. One fraction contains acid soluble pool, that is biomicromolecule. Another fraction is having biomacromolecule. But there you also see the lipids, which are not strictly biomacromolecules but still are present there. So this is the reason that I'm asking everybody. Please pay attention. Yes. Why do we have lipids there? Yes. So why is it so? Let me tell you. Dekho beta, you know that plasma membranes, yes, cell membranes, or other cell membranes have lipids. Yes or no? Have lipids. Yes or no, everyone? Please pay attention. So plasma membrane or other cell membranes, they have lipids. So when we grind the tissue, what will happen then, beta? You are going to grind the tissue. This is the lipid, this is the fat. So when we grind the tissue, then what will happen? These cell membrane, they'll be broken into pieces, right? These cell membranes are broken into tissue, and form insoluble vesicles, beta. So this is the main reason, being insoluble. So they will form insoluble vesicles. What will they form? Yes, beta. What will they form? Yes, everyone. So they will form insoluble vesicles, right? Insoluble vesicles, and they are not water soluble, so they will stay there only. Are you getting my point? So plasma membrane or other cell membranes, we know that they have lipids. So when we grind the tissue, these cell membranes are broken, isn't it? They are broken and they will form insoluble vesicles, right? या फिर water insoluble vesicles. So they will stay there in that retentate because there will be so many vesicles together, students, and they are already insoluble because all that membranes, they are broken. So that vesicles, they will stay there. Where will they stay? They will stay in the retentate. So lipids, they are not the biomacromolecules, still they are present there. So their molecular weight is even less than 800 Dalton. That's a very important point, right? 800 Daltons. Done? Okay. So now when you look at the acid soluble pool and the acid insoluble pool, so we will see that acid soluble pool represents roughly the cytoplasmic composition. So overall, acid soluble pool roughly represents, what will it represent, beta? Roughly represents the cytoplasmic composition, like whatsoever is there in the cytoplasm, right? Except without organelle. So it will represent cytoplasmic composition without organelles. So if I take out the organelles, so whatever is left in the cytoplasm, if I have to see what is present in the cytoplasm, so I can check the acid soluble pool. Is that clear? Is that clear? Sure. Is that clear? So here we will also talk about the inorganic things which are present here, right? We will also discuss the inorganic things which we are going to see in the filtrate. So this part is clear. This part is clear. Sure. So acid soluble pool roughly represents the cytoplasmic composition without organelle. Moreover, sulfates, phosphates, etc. They are not organic, they are inorganic. Sulfate, phosphate, etc. are also present in it, because we will not only see the organic things in our cell, in our living body, inorganic things are also there. So that's what you people are going to see, right? That's what you people are going to see. But ma'am, what about, right? Ma'am, what about this retentate? What do we see in the retentate? So let me write it here itself. So up to this part all clear, or do you have any doubt? Quick, everyone. Up to this part all clear, or do you have any doubt? Yes? So obviously in the acid insoluble pool, all macromolecules of cytoplasm and organelles are present, right? So here it is without organelle. So whatever macromolecules of organelle are there, of cytoplasm are there, they will be in this retentate. So all macromolecules of cytoplasm and organelles are present in it. That's what you need to know, right? So let's have a quick revision. What we discussed, beta? Puneet, what we discussed? We are talking about the analysis of chemical composition. We said we'll take the living tissue, we are going to grind it with a chemical. What is the name of the chemical? Trichloroacetic acid. So I will tell you why this trichloroacetic acid, that is also we will discuss. But first let's revise this particular experiment. So after that, what will be there? A kind of paste, a kind of slurry will be obtained, right? So now you are going to strain it. Now when you will strain it, you have to use the cloth. You can go for normal cotton cloth or the cheesecloth. So when you try to separate it, you get the filtrate or the retentate. So what this filtrate is representing? Filtrate is showing the acid soluble pool, and this retentate is showing the acid insoluble pool. So we are getting two fractions here. So there is a trick for this fraction, that is fast. What's the trick, students? The trick here is fast. That is, filtrate is acid soluble. So which acid are we talking? We are talking about the trichloroacetic acid. So this acid soluble pool, when it comes to the organic compound present in it, they are the biomicromolecule. Their molecular weight is less than 1000 Dalton. It will be somewhere in the range of 18 to 800 Dalton. Now, moreover, this acid soluble pool roughly represents the cytoplasmic composition. right? Whatever is there in the cytoplasm, that too without organelle. So sulfates, phosphates, they are also present here. And here in the retentate beta, organic compounds are there. Which organic compound? Whose molecular weight is above 10,000 Dalton. Whose molecular weight is above 10,000 Dalton. But here we also see the lipids, which are not macromolecule. They are not even the polymeric substances. They are just having the molecular weight less than 800 Dalton. But why are they present here? The reason is very simple, right? In membranes, we are having these lipids. So when we grind the tissue, we are breaking these membranes. So these membranes are going to form some insoluble vesicles. So they are water insoluble vesicles, which we are going to see here. That's what we have discussed so far. So anyone in the class who can tell me why trichloroacetic acid? Why trichloroacetic acid? Do you know trichloroacetic acid, right? Short form TCA, is a strong protein precipitating agent. Do you know that? It's a strong protein precipitating agent. Anyone? Do you know that? It's a strong protein precipitating agent. So what does it mean? It will cause the proteins to denature and precipitate from the solution. Is that clear? Yes? Yes, please. Yes, please. Any doubts? Any doubts? So beta, it's a strong precipitating agent, protein precipitating agent rather. So because it's a strong acid, right? It's a strong acid. So it's a strong protein precipitating agent, and it is overall it is a strong agent. So it will do denaturation of protein. So it will denature proteins. So ma'am, so what will happen then? So it will separate the proteins from all other compounds present there. Now moreover, you know that in the living body, enzymes are present. Almost all enzymes are protein. So when you put this particular chemical there, even the enzymatic activity will also stop. So whatever that living system is, it will be there in, it will be just like, it will be in that intact state because enzymes will not work further. So it causes the denaturation of proteins, right? And you know that most of the enzymes, they are proteins. So when you will put this particular chemical, what will happen beta? When you are going to put this chemical, that enzymes will also not work. So in that living body, in that living structure, no further chemical changes will occur. Right? No further chemical changes will occur. As Maitri is saying, right? She is a regular student, study in the batches, give proper responses. She is now, still she is reverting it, denature protein, makes it insoluble and then will precipitate out. That's why you are using this particular chemical here. Is that clear? So it's a strong acid. Is that clear, Bacha? It's a strong acid. So if you will put it, it is going to lower the pH. Okay? Okay? So denature proteins, that will become insoluble and precipitate and then precipitate out. Is that clear? And then precipitate out. So that's why you are using it. So do not just remember the chemical name, do remember why is it happening. So you can see here, again come to the NCERT, right? Let's discuss the NCERT part here. So whatever topics we are going to complete, we are going to revise that from NCERT. So you can see element analysis is performed. So if we are performing that analysis on plant tissue, animal tissue or microbial paste, they are saying you will get the similar list. Right, that we will come later. So after that, they are saying if the same analysis is performed on a piece of earth crust, right, as an example of non-living matter, right, we will see two lists beta, right, or obviously you will see carbon, hydrogen, oxygen, nitrogen in the earth crust as well as in the living body as well. We'll compare that too. So all the elements present in the sample of earth crust are also present in the sample of living tissue. Right, our composition is somewhere. So basically we are made up of same things. It's just that the quantity can change, right? The amount, it varies, but otherwise this is what we are going to see there. We'll talk about that table. So now come the how to analyze the chemical composition. So simply whatever we have read, see, we can take any living tissue. Right, student, you have to add trichloroacetic acid, right, using a mortar and pestle. You will get a slurry. You have to strain it. You will get two fractions. So one is bacha acid soluble, that is filtrate. Another one is retentate, which is acid insoluble, right? So scientists have found so many organic compounds in the acid soluble pool, right? So then they are saying In the higher classes, you will learn about how to analyze living tissue sample and identify a particular organic compound. As of now, so we can say acid soluble pool is having many organic compounds, but even if we want to separate that particular organic compound, in the higher classes, we learn about that, we study about that. This part is clear? This part is clear. So it will suffix to say here that one extract the compound, then subject the extract to various separation technique till one has separated a compound from all other. So in simple words, they want to explain that okay, fine, you made two fractions, you will have two different compositions, so you can further analyze it, you can further separate it, you can further separate various compounds from each other. But when will you study that procedures? In the higher classes. Is that clear? So we can even isolate, we can even purify a particular compound. Okay, so let's decode the complete NCERT. So now they are saying analytical techniques when applied to the compound, obviously they will tell us what? The molecular formula and probable structure of the compound also. So when you will try to check which particular compound is there, definitely you will get the molecular struct formula, isn't it? Isn't it? Now, here one more thing is mentioned, students. One more thing is mentioned. All the carbon compounds that we get from living tissue can be called biomolecules. Yes, ma'am. What is the line that we have here, bacha? They are saying all the carbon compounds. What are they saying? Please pay attention. All the carbon compounds that we get from living tissue, yes, please, can be called as biomolecules. Hanji, we can call them. Yes, they are the biomolecules, right? However, that's the important point, beta. However, living organisms, what else do they have? However, living organisms, beta, they also got inorganic compounds. Is that clear? Inorganic elements and compounds. So it is not just the organic compounds that we see in a living body, we also see inorganic compounds. Am I clear? Am I clear? So there comes the one more experiment, right? Here comes one more experiment. So let's talk about that. But before that, if there is any doubt, do let me know in the chat section. And guys, make sure that you hit the like button, and whatsoever your doubts are, yes, you can post it here. We'll definitely discuss that. So this trichloroacetic acid part clear? Yes, people. This trichloroacetic acid part clear? Sure. Yes, please. Sure. Chaliye, so let's discuss that particular experiment. Done? So, okay. So that elemental analysis part is there now. So now we will learn about that particular experiment. So first of all, again, we have to take a living tissue. What we have to take? We have to take a living tissue. Yes or no? What we have to take? We have to take a living tissue, right? It can be leaf, liver, I told you, anything you can, leaf or liver. Then beta, we have to measure its weight. What we have to do, right? Measure its weight. Its weight. Now, when you are taking a living tissue, you are measuring its weight. Don't you think that is going to be the wet weight? Yes, please. What will that be? You will take a living tissue. What will you do? You are going to, it can be any leaf, liver, whatsoever it is, plant material, animal material. Then you are going to measure its weight. But what that weight is going to be, Puneet, that weight is going to be wet weight. Why? Because you are considering the water component here, right? You are considering the water component here. After that, dry it. After that, dry it. Right now, you are going to dry it. This part is clear. Now, you are going to dry it. So, when you will dry it, what will happen? All water evaporates. Obviously, that is obvious. Do you know that or no? No, ma'am, we are not aware of it. We don't know after drying how the water will evaporate. Are you aware of it? Yes. Yes. So, after this, all the water evaporate, then burn it. This is a kind of destructive experiment. Then what will you do? You will burn it. So first of all, you will take the living tissue, you are going to dry it, all the water will evaporate, right? Then again, you can measure the weight, and now this time the weight that you will get, it is going to be the dry weight. So in the dry weight, beta, water component you are not considering. You are not considering the water component here. Is that clear? Statue of affection. Is that clear? Now you are going to burn it. So when the tissue is fully burnt, carbon compounds are oxidized to gaseous form. Right? So, when the tissue is fully burnt, carbon compounds are oxidized to gaseous form. Right, beta? Right, all the carbon compounds, what will happen to that? They'll be oxidized to gaseous forms like CO2, right? Water vapors are removed from the remnant and it is called the ash, right? CO2 and water vapors are removed from remnant. From that remaining part, all that water vapors, they'll be removed. And it is called as ash, ASH. That's what you will get. So you are going to burn something. You all have seen that, right? You all have seen that. Now, what's the next part? So look at this part. Same thing is given here. A destructive experiment has to be done. You'll check the amount of weigh a small amount of living tissue, and when you are weighing it directly, when it is having the water component, it is the wet weight, then you will dry it, all the water will evaporate, the remaining material will give the dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to, right beta, they all are oxidized to the gaseous form, CO2, water vapor, they all are removed, and the remaining is called the ash. So this ash will have what? Inorganic elements beta, like sulfate, phosphate, calcium, magnesium. So they are also found in the acid soluble fraction. Is that clear? Is that clear? So that is what we have mentioned here. So here you can see, so this ash contains inorganic compounds, right? So it contains inorganic compounds. Yes, please. First of all, inorganic elements like calcium, etc. And compounds may you have sulfates, phosphates, etc. And you know that all the things, where do we discuss these things? We have discussed that in the case of acid soluble fraction. So it will be there in the acid soluble fraction. Is that clear? So calcium and all that thing, calcium, magnesium will be there, and compounds may, just elements may you have calcium, you have magnesium. In the compounds you have sulfates, you have phosphates, etc. Okay beta, sulfate, phosphates, etc. Is that clear? Is that clear? So any doubt? Any doubt? So these are the things which are even present in acid soluble fraction. So I hope this part is clear. So acid soluble fraction is not just showing you the organic things, they are also showing you the inorganic elements and compounds. So this is what we have learned in this particular experiment. So you can see element analysis give elemental composition of living tissue. How? In the form of hydrogen, oxygen, chlorine, etc. While analysis for compound, like when you go for the elements, you will see hydrogen, oxygen. Definitely you know what is an atom, what is an element. Then when you analyze the compounds, you will get an idea, right, of the organic and inorganic compound. Then they are saying that from the chemistry point of view, right, we will check the functional group, whether it is having aldehyde, ketone, aromatic compounds, etc. Or beta, from biological point of view, we shall classify them into amino acid, nucleotides, etc., etc. So that is it, right? That is it. Now look at this particular table. What this table is saying, it is giving us the list of inorganic constituent of a living tissue like sodium, potassium, calcium. So in text they have just mentioned calcium, magnesium, but calcium, magnesium, water, right, sodium, potassium, and when it comes to the compound, you have NaCl, CaCO3, phosphate, sulfate, they all are present, right? So this is phosphate, this is sulfate, right? Do remember that. Do remember that. That's it. Okay, that's it. Now One more table. So from the biomolecule chapter, make sure you do not miss any table. Here, this table is from the new NCERT. So if you have old NCERT, here they will not show you 9.5, rather they will show you 0.5. So how can we focus on these tables, or what we have to understand from these tables? Let's discuss that. See, they are showing the earth crust, which is non-living, human body, which is living. So if I talk about the non-living, that is earth crust, what do we have there? In Earth's crust, what do we have there? Right. So I will focus mainly on bacha carbon, hydrogen, oxygen and nitrogen. So look at this. Maximum is what? Chaliye, overall, let's see the table. Maximum is what? Maximum is oxygen. Number one. Number one is what? Oxygen. Look at the composition. Then bacha silicon is there. Yes or no? Silicon is there. Then calcium is there. Sodium is there, magnesium is there, yes, right. Then you have hydrogen. After that, this carbon and nitrogen are at same level, right? We missed something? No. So overall, if I talk about the oxygen, yeah, this maximum is oxygen, carbon is also there. So maximum is oxygen. After oxygen, what do you have? You have, after oxygen, what do you have? Yes, hydrogen is there. Then beta, then we have what? Carbon and then nitrogen. So N is very little here. N is very little here if I talk about the earth crust. First oxygen, right? Then there is the hydrogen. Then there is beta carbon and then there is the nitrogen, right? Nitrogen. Overall, if I will say oxygen maximum, then you have silicon, then you have calcium, then you have sodium, है ना? After sodium, you have magnesium. Do check it. Am I writing it correctly or not? Hydrogen. So after hydrogen, beta, carbon or sulfur, they are at same level, and then you have nitrogen, very little. Overall, this is what you have. That's how you have to decode the complete NCERT. Then you have human body, that is a living body. So in the human body, if I will see, so what do we have at number one? Definitely again here. Here you have oxygen, but after oxygen, second is carbon, third is beta, third is hydrogen here, and fourth will be the nitrogen here, right? So after that, fifth will be your calcium here, sixth will be your, yes, so I'll pick that up. So carbon maximum, right? Then after carbon, oxygen maximum. After oxygen, you have carbon, then hydrogen, and then beta we have what? We have nitrogen. So that's what you have to remember. So in earth crust, it is oxygen, hydrogen, carbon and nitrogen as per this table. In the human body, it will be oxygen, carbon, hydrogen and then nitrogen. So that's how you have to remember. And in the human body, just here silicon is there. After oxygen, it is silicon, but in human body, silicon is negligible. Silicon is what? Negligible. So that's how we have to decode the tables as well. We are not supposed to miss that. Any doubts? We are not supposed to miss that too. Any doubts? Pakka? Sure? 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 Done, bacha. So that's how we have to decode the NCERT. Okay, and that's how we study properly with proper notes making and all. And in class 11th, you have to be very sincere, beta. Class 11th is very, very, very important. Okay, so these flow charts are also given. So don't worry about that. If you need the quick revision, you can see here. You have the detailed chart and then there is a simple flow chart as well. So you can see living tissue grinding in trichloroacetic acid, and then you have thick slurry strained through cheesecloth. Two fractions are obtained. So let me rectify this particular part. So yeah, so write it properly. Cl3C CuOH. Done? Okay. So any doubts? Any doubts? So two fractions are obtained. So fast is the trick. What is the trick? Fast is the trick. Done. Now the next part beta that we have to understand is we'll talk about, and this is the element analysis. dry piece of living tissue, burn the tissue, ashes left behind. So this is showing that inorganic elements and compounds. Okay, bacha, right? So done and dusted. So this table part is also done and dusted. Now next is the primary and the secondary metabolites. See, in the living body, that's what we learn, and I hope you know that there is something which we call as metabolism. Right? There is something which we call as metabolism. Metabolism is a defining property of living organisms. What's that? It's a defining property of living organisms. What is the meaning of defining property here? What is the meaning of defining property here? Defining property is something which is present in living beings without any exception, and it is not shared with the non-living beings. What is the meaning of defining property here, beta? Defining property in the case of living organisms, just say living organisms, they have characteristic feature, peculiar or particular features are present in living organisms. Now, the another category that we are using here is we are saying defining property. So when we use the word defining property, they are present in all the living beings without any exception, and it is right, it is present in all the living beings without any exception, and it is not even shared with the non-living things. So when we talk about the metabolism, that is chemical reactions, right? All the chemical reactions going on in a living body, yes, sum of all the chemical reaction, sum of all the chemical reactions in a living body is basically the metabolism, right? So if somebody is living, yeah, something is living, so being living and metabolically active, these two things are synonyms. They are having same meaning. If somebody is living, yeah, something is living, definitely metabolism will be there, definitely chemical reactions will be there. So sum total of all the chemical reactions in a living body, what do you call it, beta? You call it as metabolism. What do you call it? You call it as metabolism. So in metabolism, there are two words, anabolism or catabolism. Anabolism or catabolism. Cata, cut, to break. So catabolism, it shows breakdown. So if in living body reactions are going on, sometimes there will be a build-up and sometimes there will be the breakdown. So these type of reactions collectively, they are making the metabolism. So if metabolism is going on in our body, so we have the metabolites also. Yes or no? What do we have? We have the metabolites also. Chemical compounds that are participating in this metabolism. Yes or no? Yes or no? Definitely. 100%, ma'am. Metabolites will be there that are participating in that chemical reaction. If there is a chemical reaction, there will be the compounds which will participate there. There will be the substrates that will change into the products. So these are the metabolites. Now we have to learn a very important topic, that is primary and secondary metabolite. Primary and secondary metabolite. So that's the topic. So what are these metabolites? These are that intermediates, that compounds which are there in that living reaction, right, beta Nimish? So primary and secondary metabolite, that's what you are going to learn. So in simple words, if I talk about that, let's suppose this is a living organism, right? Multicellular, unicellular, whatever it is. There are some compounds, some metabolites rather, which are having identifiable function. What is the meaning of identifiable function? Means we know this is how it is playing the role. We can find out that they are important for the normal physiological processes. So such type of metabolites are primary metabolites. And especially in plants and fungi, we also see secondary metabolites. They do not have that identifiable function. They form during that chemical reactions, and they are obtained from these fungi and plants, and they can be used for the human welfare. So it is believed that these secondary metabolites, they form, right, for defense or such type of processes. So that's how you can relate. So when you are talking about the primary metabolites, yes, they form during the growth phase, beta, and secondary metabolites, they form during the end or near the stationary phase of growth. What does it mean? Like, let's suppose when an organism is growing, right, these primary metabolites will be there. They are formed. They have identifiable function. That's the NCERT. They have identifiable function. They are important. Like, you're talking about amino acid, you're talking about carbohydrates. Now, secondary metabolites, when the growth phase is done, when there is a stationary phase, that growth has stopped, that stationary phase is there, that maximum growth is there, the stationary phase is there, they form then. So they are directly involved in maintaining normal physiological processes. For our basic processes, for our normal physiological processes, we need them. But beta, they have no role in growth, development and reproduction. They have, right? So their role in living organism is not discovered. But yes, primary metabolites, they are having the important role there. So your amino acid, carbohydrates, they all are primary metabolite. But secondary metabolites mein, jaise if we are getting rubber, drugs, spices, oils, right, they all are the secondary metabolite. Now, with the help of this particular table, you can revise it quickly. You can even write it in normal board exams, and this topic is very important for the NEET exam as well. Okay, now from the NCERT, when we read it, so you can mark that. Okay, see this. Biomolecules are metabolites. We know that they are taking part in the chemical reactions. So in animal tissues, one notices the presence of all such category of compounds which are in the figure 9.1. So they have given this 9.1. So they are saying ki yes, acha, this is the table. They are talking about the figure beta. So it is present. Now next part is, these are the primary metabolites. However, when we analyze the plant, fungi and microbes, right, we see thousands of other compounds than the primary metabolite, right? So, jaise alkaloids are there, flavonoids are there, rubber is there, essential oil, antibiotics, colored pigments, right, scents, gums, etc. They all are what? They all are called secondary metabolites. They all are called secondary metabolites. Now, next part is, while primary metabolites have identifiable function, just now I told you, and they have important role in normal physiological processes, right beta, but the role of secondary metabolite is not that clear in that host organism, right? It is not that clear. So many of them are used for the human welfare. Is that clear? Is that clear? So this is the next important table which we need to remember, right? So this table is showing the list of beta secondary metabolites. So just say here, the very first thing that they are discussing is what? They are talking about the pigment. So now let's understand this particular part. So remember, in this particular chapter, what is important beta? In this particular chapter, each and every table is important. Okay, each and every table is important, and that's what we are also going to discuss here. Any doubt so far? Any doubt so far? So this is it is a very important topic. So pigments. In the case of plants, you know pigments are present. Just leaves are green in color, chlorophyll is there. But plant is not just green in color. Plant is not just green in color. It is having other colors also. So different type of pigments are there. So what these pigments are? They are the protein complexes. They are the proteins, right? They can capture the sunlight. Electrons will excite. We'll see. We'll discuss that in the plant physiology. They are imparting the color. They can capture the wavelength, certain wavelengths of light. Right? So now here, when you talk about the pigments, different pigments are given. Like the word used here is carotenoids. So beta, it is lipid soluble pigment, right? Carotenoids. Here you talk about the xanthophylls. Remember, xanthophylls impart yellow color. Carotenes are there. Then you have anthocyanins. Just say your petal colors, they will show the anthocyanin. So anthocyanin is water soluble. So, no doubt in the plant physiology you will read it, but overall, whenever you will make the short notes, do include this particular table and write the important pointers like this. Now, next chemical compound is alkaloid. So, alkaloid mein the very first thing that they are mentioning is morphin. Morphin is bacha analgesic. What is the meaning of analgesic? Analgesic means pain reliever. What's that? It's a pain reliever. It's a pain reliever. Okay. It helps us to get relief from the pain. It's what that technical term that we use is analgesic. That is the morphin beta. Morphin. So this morphin is obtained from poppy plant. Right? In class 12th also we read about that. Poppy plant paper. Now, when you talk about the codeine is also analgesic, and codeine we see in the cough syrup. Do check the cough syrups, right? Not in all the cough syrup, but cough syrups, they do have these codeine, right? They do have these codeine. So it is found in cough syrup, beta. Is that clear? Is that clear? Yes, everyone. So it is also opioid, like it is also obtained from that poppy plant. Okay. Okay, that is also analgesic. Done. Any doubts? Yes, bacha. Any doubts? Yes, please. So, beta, next is the terpenoids. Beta, terpenoids are also the organic compounds, right? They are mainly found in the plants, terpenoids. So, they are having isoprene units. So, regarding this, you don't need to know much. So, next is the toxins. So, toxins will include heparin or ricin. Essential oils are also there, right? And in some oils also these isoprene compounds are present. Okay, so lemongrass oil is there. Then toxins are there. So toxins mein what do you have, right? Yes, codeine is also a pain reliever, bacha. It is mainly there in the cough syrup. Okay, it is mainly there in the cough syrup. Is that clear? Is that clear? Done. So heparin and ricin, they are the toxins. Beta, they both can inhibit protein synthesis. They can stop protein synthesis. Okay. So this heparin is obtained from rosemary. This ricin is obtained from castor. Trust me, you have seen these particular plants as well. Just it is obtained from rosemary. Okay, heparin. And this ricin is obtained from castor. Ricinus is the scientific name there. So castor is also a medicinal plant. So they both are the toxins. They both can inhibit protein synthesis. So if proteins will not form, how the body or the function, how the things will work. Then students, next is the lectin. Lectin is basically concanabalin. This example is given. Beta, lectins, you can say they basically bind to the carbohydrate. Okay, what is the term that we have here, bacha? What is the term that we have here? Lectins. So lectins are what? They will bind to the carbohydrate. They are somewhere the carbohydrate binding proteins, right? Right. So they are what? These lectins are carbohydrate. What are they, bacha? They are carbohydrate binding proteins. What are they? Carbohydrate binding proteins or glycoproteins that bind to the cell surface. So they have different roles. Okay, sometimes in recognition also, but that much is sufficient. Then drugs, bacha, vinblastine is there. So this vinblastine is even anti-cancerous. This vinblastine is even anti-cancerous. Do you know that? Anti-cancerous. So then this is the curcumin obtained from your turmeric. So if I talk about this vinblastine, you people will find it from this Vinca rosea. Catharinthus rosea, Vinca rosea. It's a very commonly occurring plant and it is anti-cancerous. It is anti-cancerous, and bacha, this vinblastine, as I said, it's a very commonly occurring plant. You people have seen that. Just a minute, if I'll get a chance, I'll try to show you here. So vinblastine, you all have seen it, trust me. So it is obtained from Vinca rosea. This, I think you have seen that. Yes or no? It is Vinca rosea. Can you see that? This, it's such a common plant. So vinblastine is obtained from it. So it is anti-cancerous. Okay, it is anti-cancerous. So beta, in our batches, we are not just highlighting NCERT or writing the points. We are decoding the NCERT like that. So yes, it is periwinkle. Navya is right. Vinblastine is, this Vinca rosea is periwinkle. Excellent, beta. Yes, so that's what you need to know. And as I was saying, that in the batches, we are not just highlighting NCERT or reading NCERT. We are discussing extra points wherever required. We are also talking about the tables in such a way. So this is basically the dissection of NCERT which is going on in the batches. Okay, so that's how you have to understand the NCERT. I know some of you will be like, ma'am, what's the point of knowing this, remembering this? No, you never know. You never know. Examiner can form any type of question. We have to prepare. We have to prepare for the worst. That's what I believe. Okay. So from this particular table, what sort of question do they ask? Sometimes, bacha, they ask you the match the column type of questions, right? Match the column type of question, like they'll give you the list. Just say, let's suppose here I'm writing toxins, okay, and drugs, alkaloids, pigments. I'll give you one example, okay? Toxin, drugs, alkaloid, pigments. Now here I am writing heparin, I'm writing vinblastine, I'm writing lemongrass oil, sorry, I'm writing anthocyanin, and then I'm writing what? I'm writing codeine. So here I'm writing, yes, 1, 2, 3, 4. And here I'm writing A, B, C, and D. So match the column, bacha. Match the column, quick. Quick, students, match the column. Quick. Yes, please. I would like to see the answers in the chat. Yes, bacha. So you know that. How can we match it? I'm not giving you the option. You can simply tell me this one will go, toxin will go with heparin. The drug will go with this vinblastine. Alkaloid, codeine, pigment will be anthocyanin. So anthocyanin is a water soluble pigment. Carotenoids are lipid soluble pigments. So that's how beta you have to decode the NCERT. Polymeric substances is Shima. Rubber is there, gum, cellulose. Right, so we'll learn about it, we'll get to know about it. So this is about the primary and the secondary metabolite. And now what are we going to discuss? We are going to talk about the carbohydrates. So today itself in the batch, bacha, today itself in the batch, I was discussing this with the students. You can just simply check the PDF of class 12th biomolecule NCERT. You people are going to find, that things right there where they're talking about monosaccharide, oligosaccharide, polysaccharide reactions are there, right? The Fisher projection, the Haworth projection, how you are making the ring, all the things are discussed here. So prepare that chapter accordingly. Prepare this particular chapter accordingly. So biomolecule from class 11th, cell cycle from class 11th, right? Even the basics of your plant kingdom or morphology, they are very important for your class 12th. They are very important for your class 12th. Are you getting my point? So let's revise the carbohydrates. Now, carbohydrates, what's the word? Carbohydrates, hydrates of carbon. What does it mean? We'll discuss that. Hydrates of carbon, carbohydrates. Right? What is the term? What is the word that I have used? I am saying they are the hydrates of carbon. So it is the first or the main respiratory substrate. Whenever we eat something, oh, sorry. So main respiratory substrate. So bacha, we eat something, right? We eat the food. The complex food is converted into the simple form. You know that will be glucose. So glucose will be oxidized. This is what we study. No doubt it is the assumption. We assume that it is the glucose which is the only the respiratory substrate. But still, if we have to understand that process, what are we going to discuss? So what is happening basically? The carbohydrates, we are saying they are the main respiratory substrate. You say glucose is the example. You all are well aware of this term, that is glucose. So glucose, it will be oxidized in the presence of oxygen. It will break in the presence of oxygen in multiple steps. The energy will be released. That energy is captured as ATP, right? That's how our system it works. Okay. That's how our system it works. So that's what you need to understand here. So what are we talking? We are talking about the carbohydrates. They are the main respiratory substrate, your main source of energy. So if we talk about them, so they are having carbon, hydrogen, oxygen with the ratio of, so carbohydrates have, the carbohydrates contain carbon, hydrogen and oxygen with the ratio 1:2:1. So basically, what am I trying to tell you? Ye dekho, just like water, just like H2O, let me show you. You have carbon, you have hydrogen, you have oxygen in the carbohydrates. What do you have in the carbohydrates? Everyone please pay attention. Hello, wake up. You are not allowed to sleep. Let's finish it first. So you have carbon, you have hydrogen, you have oxygen. So it is 1:2:1. That's what we have. 1:2:1. So basically, look at this. 1 part of carbon, 2 parts of hydrogen, 1 part of oxygen. Yes or no? 1 part of carbon, 2 parts of hydrogen, 1 part of oxygen. So look at the hydrogen and oxygen here. 2 parts of hydrogen, 1 part of oxygen, just like water. So that's why they are also referred to as hydrates of carbon. That's why they are also referred to as hydrates of carbon. Is that clear? Is that clear? Yes. Yes, please, bacha. Is that clear? Sure? Sure? So carbohydrates, how do we define it, bacha? So carbohydrates, if I talk about their proper definition, so what do we say? So carbohydrates are, yes students, carbohydrates are poly means many, polyhydroxy, that is OH, polyhydroxyaldehyde or polyhydroxy ketone. I'll give you the example, bacha. Polyhydroxyaldehyde or polyhydroxy ketones, or the compounds on hydrolysis, or the compounds which on hydrolysis yield them. Are you getting my point? So they are chemically what? Chemically they are what? So carbohydrates are chemically, carbohydrates are what? Chemically carbohydrates are polyhydroxyaldehyde or polyhydroxy ketones, and the compounds the compounds which on hydrolysis, and the compounds which on hydrolysis give such substances. So that's how you define it. Done, bacha? That's how you define it. Right? So and the compounds which on hydrolysis give या yield such compounds. Right? So carbohydrates are polyhydroxyaldehyde or polyhydroxy ketones, and the substances which on hydrolysis yield such compounds. One second. Yield such compounds. Okay. So bacha, I'll give you the example, and that's the most common example. I believe you all know glucose. Glucose is a carbohydrate. Okay. Glucose formula is C6H12O6. What is the formula of glucose, bacha? C6H12O6. Okay. How do we make it? Ye dekho, C double bond OH. That's the structure of glucose, and it is the chain form. It is also known as Fischer projection. So please pay attention. So 1, 2, 3, 4, 5 and 6. So that's what we have. Look at this. This is the glucose, right? This is the structure and the formula of glucose, C6H12O6. I think you all know that. Yes or no? I think you all are well aware of it, right? C6H12O6. So look at this. See, what's that? It is functional group. Which functional group? This is aldehyde. Aldehyde. C double bond OH. This is aldehyde. Right, beta? That's the functional group. Now look at this. Look at this structure. It is having many OH, 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 OH. Do we have OH here? Yes, ma'am. So polyhydroxy aldehyde. Are you able to understand? Polyhydroxy aldehyde. What's the word, bacha? Polyhydroxy aldehyde. So which functional group is there? It is aldehyde, C double bond OH. So that's the functional group that we have. That's the functional group that we have. Yes, bacha, any doubt? Any doubt here? So it is polyhydroxy. It is polyhydroxy aldehyde. Am I right? It is polyhydroxy aldehyde. We'll give you one more example, that is fructose. So fructose is basically the isomer of glucose. The formula is same, C6H12O6. Now here what you people can see, look at this. This is your fructose. What's that, bacha? It is fructose. So this one is the glucose here, right? This one is the glucose here, C double bond OH, and look at this, many OH group, and this one is the fructose here. So in the fructose, this is one is the functional group. Bacha, this functional group is ketone. Ketone. Which functional group is there? Ketone. So ketone is a sub-terminal functional group. It will not be used at end. Just say aldehyde is a terminal functional group. You'll use it at the end. It will be here, right? It will not be there at the end. So this is ketone, right? So ketone, C double bond O, this. That's your functional group here. So glucose formula is C6H12O6. Fructose is also C6H12O6. So basically fructose is the isomer of glucose. I hope you know that, right? So their basically their formula is same, but the structure is different. So glucose is the example of aldose, aldehyde. It is the example of a ketose, right? It's a ketone, it's a aldehyde. So look at this, so many OH groups are there. So it is justifying that polyhydroxyaldehyde, polyhydroxy. And if the functional group is aldehyde, you will say polyhydroxyaldehyde. If the functional group is ketone, so you will say polyhydroxy ketone. So up to this part all clear? Up to this part all clear? Any doubts? Any doubts? Pakka? Sure? Sure, bacha? Done? So carbohydrates, that's what we need to understand now. So simple carbohydrates which are soluble in water and sweet in taste are sugars. So we will now classify them, and then accordingly we have to discuss it. So if any doubt, please do let me know. Right, beta? Okay. So if the carbohydrates, they are soluble in water, they are sweet in taste, then you use the word sugar. So you cannot say that all the carbohydrates, right, they are sweet in taste, not at all, right? So let's classify these carbohydrates on the basis of number of saccharide unit, number of sugar units. So we will talk about this monosaccharides, oligosaccharides, or the polysaccharides. So that's how we are going to learn that. So you make sure you revise it. Right, so in the biomolecule chapter, that's the rule, bacha. वैसे तो it is the rule for all the classes, but overall for biomolecules, I'll tell you that always make sure that you revise it. Right, do not go for the back, do not basically miss any lecture, attend the lectures live, and after the class do revise it. Okay, do revise it. Done, bacha? So let's talk about the classification of carbohydrates. So it is on the basis of number of saccharide units. So carbohydrates on the basis of number of saccharide, that is sugar, on hydrolysis. Okay, on hydrolysis. So chaliye, let's discuss that. So there are three things which we need to focus. Monosaccharides are there, bacha. Oligosaccharides are there. Monosaccharides are there. Oligosaccharides are there, and then comes the polysaccharides. Is that clear? Monosaccharides, oligosaccharide, and polysaccharide. So in your class 11 NCERT, they have directly mentioned, right, bacha, polysaccharide. What they have mentioned, bacha? They have mentioned polysaccharide, but if you really want to understand polysaccharides, you need to know what monosaccharides are. So mono means single, saccharide means sugar. So bacha, these are the single sugars which on further hydrolysis will not give us the sugar. Rather, when we will break them, we will get that carbon hydrogen, isn't it? So they are the simplest sugars which cannot be further hydrolyzed. So what these monosaccharides are, they are the simplest sugars which can't be further hydrolyzed. Yes, please, which can't be further hydrolyzed. So this is what we have here in the monosaccharides. Okay, now in the oligosaccharides, in detail we will discuss that. So they have, so number of sugars in oligosaccharides, yeah, number of saccharides in oligosaccharides will be from 2 to 10. That is, just say we will have, if 2 sugars are there, this is going to be disaccharides. If 3 sugars are there, trisaccharides, and so on. So in oligosaccharides, that's what you are going to learn. That is what you are going to understand. No. Hishima, oligosaccharide, it doesn't mean 2 saccharides. No. Oligosaccharides mein number of saccharides can be from 2 to 10. It can be from 2 to 10. In monosaccharides, it is only one, bacha. It is only the single sugar, right? In monosaccharide, what is it? It is only the single sugar. Okay, it is the simplest sugar which cannot be further hydrolyzed. Just glucose, C6H12O6, right? That's a monosaccharide. Now in the oligosaccharide, the number of sugars can be 2 to 10. So if 2 sugars are there, you will call it as disaccharide. Di means 2. Just say for an example, you will talk about the sucrose here, you will discuss the lactose here, you will discuss the maltose here. So if there is the trisaccharide, so trisaccharide, it will be having 3 sugars. It will be having 3 sugars, and then comes the polysaccharides. Poly means many, right? Poly means many. So here it will be more than 10 sugars, right? And just now we have discussed that they'll be found in acid insoluble fraction. Am I right? Where are we going to find it, bacha? We are going to find it in acid insoluble fraction. Is that clear? Is that clear? Done? Is that clear? So let's start from the basics here. And what are we going to discuss? We are going to talk about the monosaccharides. Okay, first of all, we will talk about the monosaccharides. Ready? Yes. So up to this part, any doubt? Yes, please. Up to this part, any doubt? Pakka? Sure? So let's talk about the monosaccharides, the simplest carbohydrates which cannot be further hydrolyzed. And now here you'll see general formula. First of all, we will see general formula. No doubt we will also study the exceptions from general formula. So it is going to be CnH2nOn, or you can also write it CnH2ON. That's what you have. That's the general formula. Is that clear? Is that clear? So they are which are sweet in taste. So if I talk about the properties, number 1, they are sweet in taste. Yes, monosaccharides are sweet in taste. Bacha, they are soluble in water as well. So they are sweet in taste. Yes, please. They are sweet in taste. They are soluble in water, and they are highly soluble because they have many OH groups. Right? Yes, please. Now, they exist in open chain form. They exist in open chain form and in the ring form या फिर cyclic form. That is also what you are going to see here. Right? They are optically active. Optically active means can rotate plane polarized light. Can rotate plane polarized light. Don't worry, I will discuss each and everything. The meaning of plane polarized light. Right, we will discuss everything. Right now, you just write down. Okay, so they are optically active. They can rotate plane polarized light. Okay, then bicha, they are. Yes, done? Any doubts? Okay, so next part is they are basically reducing sugars. Reducing sugars. So this is a very important point. So most of the monosaccharides, right? So reducing sugars they are, that's the word, right? So all monosaccharides are reducing. All monosaccharides are reducing sugars as they have free aldehyde or ketone group in their chain or ring form. Like this is what they have. Now, I'm sure you are not aware of it. We'll discuss that they can, reduce the Benedict solution, Fehling solution. So we will talk about that in chemistry. You people read about it, right? So they can reduce that particular reagent. So we will discuss about that. Just a Benedict test is there, Fehling test is there. That will helpful for checking the presence of glucose in urine. So one by one we will discuss that. But tell me, up to this part all clear? Up to this part all clear? Sure, bacha? Sure? Yes, please. Sure? So up to this part all clear? Yes, Hishima is right. They have free aldehyde or ketone group. So we will discuss which one is the ring form, which one is the, this straight chain form. We'll talk about that also, but up to this part, if everything is clear, do let me know. And if there is any doubt, you can ask me. You can ask me. Sure. Then we will discuss everything, right? What is the meaning of plain polarized light? What is the meaning of being optically active? We will also discuss, right, what is the dextrorotatory, levorotatory. We will discuss all the things, okay? But it is simple. This is the monosaccharide. Their general formula is this. They are sweet in taste, soluble in water. Two forms are there. This is cyclic form, या ring form is there, and they have open chain form, right? Cyclic form या ring form is there, they have open chain form. I will discuss that too, don't worry about that. Optically active and reducing. So these three things we will discuss later, but first let's talk about the classification. After that I will discuss that. So classification of monosaccharides. How do you classify the monosaccharides? So classification of, yes bacha, classification of monosaccharides is on the basis of number of carbon. Is on the basis of number of carbon. So on the basis of number of carbons you divide monosaccharides. Monosaccharides are single sugar, bacha. Monosaccharides are single sugar. But how do you classify them? On the basis of number of carbon. Just for an example, you are talking about the brick. Let's suppose one brick is 2 centimeter, 3 centimeter, 4 centimeter. So you have, let's suppose 5 different type of bricks. So one is 3 centimeter, another is 4, another is 5, like that. So you have one brick only, right? It is not like a wall or something that you are discussing. You are talking about a basic subunit only. But here what will you see? Just whether it is having 3 carbons, yeah, 4 carbons, 5 carbons, like that. So this is what you have. So classification of monosaccharides on the basis of number of carbons. So here beta, number of carbons in monosaccharide, they'll be, number of carbons in monosaccharides, they are going to be 3 to 7. Do not confuse it with the oligosaccharide. Number of carbons are going to be 3 to 7, right? 3 to 7. Already told you about the functional group, hai na? Already told you about the functional group. Just you will be having aldehyde or you are going to have ketone. That's what I told you. Now imagine if carbohydrates, yeah, if your monosaccharide, if it is having bacha aldehyde, so you will put it in, if monosaccharide contain aldehyde, you will call it as aldose. What are you going to call it? Aldose. So if it contains ketone, then you will call it as ketose. What will you call it, bacha? You will call it as ketose. So you have aldose and you have ketose. You have aldose and you have ketose. Is that clear? What do you have, bacha, in the monosaccharide? You have aldose or you have ketose. Any doubt? Any doubt? Aldose or ketose. Now, I told you the carbon number is going to be 3 to 7. That's what I told you. Yes, bacha, the carbon number is going to be, hello, good morning. Okay, so number of carbon, aldose, or here we are going to talk about ketose. So let me tell you. So if number of carbon, if carbon number is 3, bacha, it will be triose. If carbon number is 4, 5, 6, 3 to 7 na, 7, 4, 5, 6 or 7. So triose, tetrose, pentose, hexose, and then comes bacha heptose. Am I clear? Please pay attention. Am I clear? Hello, wake up. Am I clear? So triose means 3 carbons are there, tetrose means 4 carbon, pentose 5, hexose 6, and heptose mane 7 carbon are there. So aldose. So the example here is glyceraldehyde. What is the example? Glyceraldehyde. So glyceraldehyde, what do you have here? 3 carbons and the functional group is aldehyde, right beta? So the functional group is aldehyde. So you are calling it as the Yes, please. Triose, because number of carbon is 3. Yes, number of carbon is 3. Yes, please. Am I clear? So glyceraldehyde. So glyceraldehyde. So it is aldotriose. And here you have the example of dihydroxyacetone. But beta, this one will not be optically active. We'll come to that later. Just here, you can see the example is erythrose. Carbon number is 4. Here, bacha, the example is erythrulose. So this ul you have to add. Then here you have ribose, you have xylose, here you have ribulose, and here you have xylulose. There are many examples. So I'm just trying to show you this thing. Dekho, ribose, it's a pentose. Ribose, it's a pentose, means it is having 5 carbons. Ribulose, ul is the word here. In the nomenclature, you have added ul. So it is indicating the functional group is going to be ketone. There's a glucose, galactose, they are the example of aldoses. Here you have the best example, fructose. Here you have pseudo haptose, and here what do you have? You have pseudo heptulose. Heptulose. Is that clear? Is that clear? Done, bacha? Sure? Pakka? Pakka? Any doubt? Any doubt? So this ul thing and all clear? Sure. So simple monosaccharide. You start with the triose first, isn't it? You start with the triose first, isn't it? Any doubts? Any doubts? Sure. Any doubts? Okay, that's great. So you can even write it like this, and the way I showed, that is also fine. So now next part that you can do here is apply general formula. Just I told you, bacha, the general formula is what? One more example. How can I forget? So deoxyribose is also there. Deoxyribose is also there. Now general formula. So when we talk about the general formula, I told you it is CnH2nOn, or you can also write it like this, CnH2O whole square n. Now beta, if carbon number is 3, what will be the formula? C3H6O3. Am I right? C3H6O3. Am I right? That is going to be the general formula. Just for an example, let's suppose if they have given you the question, formula of ribose. Now, if you do not remember the formula of ribose, what will you do? What will you do? You know that it's a pentose. So pentose means it is having 5 carbons. N is 5 here. So you can simply put that. So that is going to be the formula, yes or no? That is going to be the formula, yes or no? Yes or no? Right? Yes. So this is how you can even figure out the formula. So now I will write one important point here. We will elaborate it later. So deoxyribose. It's a monosaccharide, it's a pentose. Its formula is C5H10O4. It's not HO5, it's C5H10O4. It doesn't obey general formula. It doesn't obey general formula. Am I clear? It doesn't obey general formula. So this is an exception. It doesn't obey that general formula. Is that clear, bacha? Is that clear? Sure? Sure? Any doubt? Sure? Okay. Now, bacha, so after this, so here, dekho, this is the, this slide I will delete. Okay. So you can see it is aldotriose, mane functional group is aldehyde. Functional group is aldehyde, right? And it is having the 3 carbons. This is glyceraldehyde. I'll tell you the meaning of this capital D, small d as well. Have patience, okay? Have patience. Then see this aldose tetrose. So this is erythrose here. This is ribose here. Look at the formula. So 1, 2, 3, 4. 1, 2, 3, 4, 5. 5 carbon. HOH, HOH, HOH. So this is the ketose that they are showing. Which ketose are they showing? They are showing the fructose. And here you have the glucose. Is that clear? There you have the glucose. Is that clear? Is that clear? Sure, bacha? Sure. So look at this aldehyde group. So aldehyde group is what? Aldehyde group is terminal. Aldehyde group is what? Terminal. Ketose group is what? Bacha, this group is subterminal. This group is subterminal. I hope this part is crystal clear. Now, there are two things that we are going to discuss. जैसे ये देखो, C double bond O H. So 1, 2, 3, 4, 5 or 6. What is it? देखो, glucose, fructose, galactose, ribose. Remember them. That's it. Rest everything you will manage. Okay. So what's that, beta? This is glucose. This is glucose. How many carbons are there? 6. है ना? Even try it. Fill it in the general formula. You can cross verify that too. C6H12O6. That's what your glucose is. But this is the open chain form of glucose. What's that? It is the open chain form. So it is also known as Fischer projection. This is the term that you use here. This is also known as Fischer projection. Open chain form. Now, when I talk about this is also glucose. If you remember it, very good. If not, you should. This is also glucose. So there are many if buts that we will discuss now. So this is also glucose. So it is the cyclic structure. It is the cyclic structure. Yes, beta. So it is known as Haworth projection. That's the terminology that you use here. And in detail, you will read about it in chemistry. In detail, beta Anas, you will read about it in chemistry. So this is also glucose. And here in this case, right? So this is going to be your carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. Right, beta? So this carbon is your anomeric carbon, ya fir we can also call it as chiral carbon. Chiral carbon, right? Which is having different groups at all sides. As a carbon, you know that 4 bonds it will make, so it is having different groups at all 4 sides. Are you getting my point? So this is the chiral carbon, right? Chiral carbon, anomeric carbon. So this ring-like structure is known as Haworth projection. So we have to talk about it, beta, and then we'll discuss you know, what is that plane polarized light, what is that D, plus, minus, that's what we are going to discuss. So let's discuss that. Okay, so any doubt so far? Please tell me, any doubt so far? Any doubt so far? Sure? Sure? Now, bacha, if you look at the groups that we are talking, even if I talk about aldehyde, what do I have? I have this na C double bond OH. Even if I talk about ketone, what do I have? C double bond O. So C double bond O, what do you call it? I hope you remember C double bond O. It is carbonyl. That's what you call it, C double bond O, carbonyl. So both these, this is a PYQ, this is a previous year question. So both these functional groups, what do they have, bacha? Yes, please. What do they have? They have the carbonyl group in it. That's a PYQ. Now, in solution, what will happen? In solution, carbonyl group, this carbonyl group in solution, what will happen? Please pay attention. And as I said in chemistry, right, beta, reducing, non-reducing, everything will be discussed. First, let's talk about that. right? First, let's talk about that. Please pay attention. First, let's talk about this particular part. Let's discuss what is this Haworth projection. There are few structures which we have to understand. We'll also learn what are the epimers, what are the anomers. After that, I will tell you the meaning of this reducing and non-reducing sugar and why we are saying dextrorotatory, levorotatory. We will discuss that. But you better focus here, then bacha, everything will be crystal clear, okay? So, Haworth projection we are discussing, which is the ring form. So, in solution, this carbonyl group, right? So this carbonyl group of monosaccharides react with. So in chemistry, you will understand it in more depth. It reacts intramolecularly. Matlab not with something else, within that. Molecularly with OH and form cyclic structure, mane ring-like structure, right? So this is going to form ring-like structure because one of its C double bond O, it will react with the OH there, right, bacha? It will react with the OH there. Is that clear? Yes, please. Is that clear? Hello? Is that clear? Sure? Sure? So it is the cyclic structure, okay? So I'll give you just one example, but we do not have to, remember that here. So just C1 aldehyde of glucose reacts with fifth OH and form ring. Right, and it is going to form ring. So that's how in the solution they react, beta, right? In the solution they react, and that's how they are going to form the ring-like structure. Is that clear? So that ring will be, right, beta, that is going to be the pyranose ring. So there are two rings that we have to understand or we have to learn here. So any doubt so far? Any doubt so far? Pakka? Sure? Are you sure? Are you sure? Sure, bache? So beta, there are two types of rings that you will see, right? So there are two types of rings that you will see. Just say one is this. So this ring is having, it is pyranose. Which ring it is? It is pyranose. So it is having 5 carbon plus 1 oxygen. 5 carbon plus 1 oxygen. You can see that. This is pyranose. Ye dekho. Look at this. Now here in this ring, bacha, this is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. 5 carbons are there, but how many oxygen do we have? We have single oxygen. How many oxygen do we have? We have single oxygen. So this 6-membered ring is pyranose, right, beta? This 6-membered ring is pyranose. So this is one thing that we have to understand. So ye dekho, in the open chain form, the C1 aldehyde of the glucose react with the 5th carbon OH, right, with the with the 5th COH and form a ring. So when you look at this O, this O is of that 5th carbon, bacha, and this carbon C1 is referred to as anomeric carbon here. So anomeric carbon, C1 here is referred to as anomeric carbon. So this is the information here. This is the word that we are going to use again. So do remember it. So what's the word that we have here, bacha? It is the anomeric carbon. Which carbon it is? It is anomeric carbon. Carbon that was the carbonyl carbon in the open chain form. So this carbon was carbonyl carbon in open chain form. Just say in open chain form, let me show you. Here dekho, this is the open chain form. So C1 with this 5th, so they are actually going to react. Okay, so this carbon here, so this carbon here, you are talking, right? So C1 and C5 they join. C1 and C5 they join. Is that clear? Is that clear? Sure? Sure? Yes or no? So in this chain, in this open chain, जैसे ये देखो left side, the OH is on left side. So whenever you will write it in the ring form, it will be up. So if OH, so it is on the left side, है ना? So if the OH is on right side, so where will you write it? You will write it down in the ring form. So you can compare. You can compare. So if OH is on the left side in the cyclic structure, it will go up. If OH is on the right side in the cyclic structure, it will go down here. So this part is clear. This part is clear. Sure. This part is clear, yes or no? One type of ring is clear or not? One type of ring is clear or not? So anomeric carbon, right? ये देखो anomeric carbon, I told you anomeric carbon because it is representing that C double bond O carbon in that open form. So anomeric carbon is not always chiral. Chiral mane which is having 4 different groups. On always chiral, but usually it is chiral. Right? But usually it is chiral. So chiral carbon, what is the meaning of chiral carbon? Which is having different groups at all 4 sides. Is that clear? So these two concepts, are they clear? So chiral carbon is the carbon attached to 4 different groups. Is that clear? It is attached to the 4 different groups. Or anomeric carbon, beta, this is the term that you will use for the carbonyl carbon in open chain form and become attached to the OH in the ring. So, right, so anomeric carbon you will use for this carbonyl carbon. Okay, in the open chain form, and in the ring also you know that, right, they will join. So this one is your anomeric carbon. So chiral carbon mane it will have all the 4, right, all the 4 sides will have 4 different groups, bacha. All the 4 sides will have 4 different groups. So anomeric carbon is not always chiral, but usually it is chiral. So this terminology clear? This terminology clear, yes or no? Pakka? Sure? So C1 is the anomeric carbon here. Done? So anomeric carbon you will identify by its origin because in the straight chain form it has to be in that carbonyl carbon form. Okay? Okay? Right? So chiral carbon or anomeric carbon, they are 2 different terms. Is that clear? These are 2 different terms. Chiral carbon mane it is having 4 different groups at all 4 sides. Anomeric carbon mane initially, just when it was in the open chain form, this carbonyl carbon is actually the anomeric carbon. But here C1 is anomeric as well as chiral. ठीक है, मतलब all the 4 sides have different groups, plus it is anomeric. It is showing that yes, I am the one who was having that C double bond O in the ring form. So this is the first part. Now we have to discuss this pyranose ring. Clear, bacha? This pyranose ring clear? Sure? Sure? This pyranose ring clear? So there are 2 very important examples which we need to discuss from this part. So this part is clear? Hello? So furanose that resemble furan. So second type that we have to discuss is furanose that resemble furan. So furan basically, yes, this is what you have. So I will give you the example. Just say this is the example of your ribose, right? Ribose. So ribose is what? 5 carbon containing sugar. So this example, this ring that you see, it is ribose. So this type of ring having 4 carbon plus 1 oxygen, that is your furanose. Is that clear? 4 carbon or 1 oxygen, that is your furanose. Is that clear? So it's a 5-membered ring, bacha. It's a 5-membered ring. So here carbonyl group will react with the OH. Okay. Done, bache? Sure? 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 Yes or no? Any doubts? Any doubts? Any doubts? So this is your ribose. Yes, beta. So here this carbon will be your anomeric carbon. Right? So now you know anomeric carbon. What does it mean? So this is the anomeric carbon here. So for the furanose, once again, I would like to repeat this part. So furanose is the word, bacha. Furanose is the word. So as I said, 4 carbon plus 1 oxygen in the ring. 4 carbon plus 1 oxygen in the ring. So this structure that I am showing you here, it's a pentose, bacha, and it is the example of your ribose. So you know that in RNA we will talk about it. We really need to understand that. So right now I'm not telling you anything as alpha beta. First we will discuss this particular part because it is important. Am I clear? Am I clear? So whenever you are talking about furanose, so if you take the example of aldose, है ना, मतलब when the functional group is aldehyde, जैसे now I'm giving you the example of ribose. So how that furanose will form in their case? माने ultimately that reaction will occur ना. So C1 aldehyde plus C4, right? C1 aldehyde plus C4. That's what you need to remember. मतलब in the case of this aldose, C1 plus C1 aldehyde, या फिर you will say C1 C double bond O bond plus C4th OH. Right? Just say if I have to make this, let me show you. That's your ribose. Okay. પાંચ. Yes. So the bond formation in between C1 or C4. Right? C1 or C4. That's how the bond formation will be there. Right? That's how the bond formation will be there. Is that clear? Is that clear? Is that clear? Sure? Sure, beta? Now, if same example I will give you for the ketone, right? Ketone, let's take the example of fructose. So, fructose may you know that it's a sub-terminal group. It's a sub-terminal group. We'll have a quick revision also, no need to sleep. So please wake up. So here it will be in between this C2 and C5 OH. Right? So this C2 and C5 OH, beta, they are going to join. Is that clear? They are going to join. Is that clear? Is that clear? Pakka? Sure? Sure, bache? Sure? So anomeric carbon will be, you will see that in the case of aldehydes, the anomeric carbon is always going to be your first carbon. But in the case of ketose, you will say it will be the second carbon. So now let me draw the fructose structure here. So I told you, remember the structure of glucose, fructose, right beta, deoxyribose will be easy then. And these are the structures only that you need to remember, rest everything will be easy. So let me draw the fructose structure here and then we will discuss it further. Just say it will be CH2OH, OH. So this is your carbon number 1. This is your carbon number 2. Carbon number 3, OH, H. I told you, if OH in the straight chain form is on the left side, it will be up here. Then again H, OH, and then it is having CH2OH or H. So you people can see that's your fructose. So 1, 2, 3, 4, 5 or 6. Please look at the structure. Please look at the structure. Kindly look at the structure here. So aldose में carbonyl group is C1, that will become anomeric. Ketose में beta carbonyl group is at C2, so C2 will become anomeric. So it is a ketose. So this C2 is anomeric carbon. So anomeric carbon, you know that what does it represent. So if somebody is still having any doubt, you people can ask me that. Right? You people can ask me that. Okay. So in aldoses, C1 carbon is anomeric. जैसे in the ribose you have seen, in the glucose you have seen, and in ketosis, C2 carbon is anomeric. Done, bacha? So anomeric carbon, which is basically that C double bond O carbon in the open chain form. Okay, so we will have a quick revision of that, and then we will learn the bond formation. Okay. Sure? Sure? Sure, bache? Hello? Sure? So let me, okay, let's revise it again. So this type of structure is the ring form, and here you can see carbon number 1, it is having that aldehyde group. So whenever I will change this ring form into the cyclic form, right, so this C1 and we know that the C5, they are going to join, the reaction will occur. So this O here, this O is actually, right, it is showing that. So it is the pyranose, having 5 carbon, 1 oxygen, 5 carbon, 1 oxygen. This ring form is pyranose, 5 carbon, 1 oxygen, 5 carbon, 1 oxygen. I hope the doubts are clear. So C1 is the anomeric as well as chiral. So वैसे anomeric and chiral, these are two different things. So anomeric carbon is not always chiral, but usually it is chiral. So anomeric carbon क्या है बच्चा? What is the anomeric carbon? So you people can see here, right? So this anomeric carbon, it was a this carbon was carbonyl carbon in open chain form. So whatever is the carbonyl carbon in open chain form, you will call it as anomeric carbon. You will call it as anomeric carbon. So in solution, bacha, carbonyl group of monosaccharide react intramolecularly with OH and form the cyclic structure. जैसे aldoses में C1 or C5, that will react. Right, beta. Here also you can see in ribose also it is C1, or for furanose, it will be, it is having 4 carbon, 1 oxygen in the ring. So C1 or C4 will react. Okay. Okay. So that's what we have discussed. So this is the structure of fructose. So you need to remember that. So fructose is a ketose. Here the functional group is ketone. So it is the second carbon, which is the anomeric carbon here. It is the second carbon, beta, which is the anomeric carbon here. So I hope up to this part, everything is crystal clear. So still, if there is any doubt, do let me know. Sure? Any doubts? Any doubts? Chali, so the next part that we have to understand is anomers, right? Next topic that we have to. So these are the basics, bacha. If basics are clear, then everything will be easy. Trust me. So if there is any doubt so far, do let me know. Yes, please. Done, bacha? So this portion is clear. So these diagrams, ye dekho, alpha glucose, beta glucose is given. Now alpha beta thing is what we need to understand, and that's a very common mistake that usually students make. So do understand that. So anomers, that's the word we are going to understand. Beta, they are the type of stereoisomers. Hello, focus. So anomers are what? Anomers are also the type of stereoisomers. In their case, the position of H and OH change at anomeric carbon. That's the word that you have to understand, right? Just for an example, first I will give you a simple example, then I will write the definition. I am writing this particular thing. HOH, HOH, OH, H, HOH and then HCH2OH. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. Now look at this in a simple way. If you have to understand, hydrogen above, that's how I remember, H above, so it is alpha form. It is alpha form. So I will call it as alpha glucose. Right, bacha? Now, so HOH, HOH, OH, H, HOH, H, CH2OH. Yes, now what's that? Sorry. OH, H. Now, hydrogen below. That's a way to remember that. So, this is beta glucose. So, already I told you that in this glucose or in the aldoses, it is the carbon number 1. It is the carbon number 1 which is the anomeric carbon. So, which carbon is anomeric? Anomeric carbon is that C double bond O carbon. In the open chain form that was having that C double bond O. So, this is the anomeric carbon here. So, if hydrogen above, it is alpha. If hydrogen below, it is beta. So, just say alpha form, beta form, alpha form, beta form, very common here. So, we need to understand it in the chapter biomolecule. We need to understand the same for the molecular basis of inheritance as well, right? Alpha and beta. So, they are known as anomers, right? So they are the kind of stereoisomers. But when I'm using the word isomers, you know that they are having the same formula, right? Isomer is same molecular formula, but structure vary. That is what we say now. When we say isomers, what do we say? Isomers mane they are having the same formula, but structure can vary. Now these are the stereoisomers. So in their case, what will vary, bacha? Their 3D arrangement will vary. Are you getting my point? Are you getting my point? So in the case of stereoisomers, what will be their molecular formula is same, right beta, but 3D arrangement will be different, right? Just say you have your left and right hand, right? Same parts are there, but their order is different now, their 3D arrangement is different now. That is what we have, right? That is what we have. Just say we have the right hand or the left hand, everything is same, but that arrangement is different. So that's what you discuss here, bacha. That's what you discuss here. Is that clear? Is that clear? Is that clear? Hello. So anomers are special type of stereoisomers, right? So what are these anomers? So these anomers are special type of stereoisomers, right? They are found in cyclic carbohydrates, माने chain form है ना, found in cyclic carbohydrates. Can you see that? Alpha beta forms found in cyclic carbohydrates. Yes, beta. So they differ only in the configuration around anomeric carbon. So they differ only in the configuration around anomeric carbon, है ना? So in the fructose that will be C2, do remember, around anomeric carbon, right? That HOH arrangement will be different, okay? Okay, so this anomeric carbon is nothing. This anomeric carbon, which is the carbonyl carbon, so now I think you guys are not going to make mistake here. So carbonyl is that C double bond O. So it is the carbonyl carbon that becomes new chiral center, that becomes new chiral center when sugars when sugar form cyclic structures. Simple. Okay, bacha. So these are what your anomers are. So yes, you should know it. So in alpha, right, the OH will be in opposite direction to that CH2OH. So you can see that. Okay. Okay. Yes, bacha. Okay. Sure. Right. Okay. Chalo. So one more structure that we need to understand here is, ye dekho bacha, ribose. It's in furanose form. This is HOH, 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 and CH2OH or H. That's what you have. So you people will see here 1, 2, 3, 4, 5. So it is alpha ribose. है ना? So in the case of beta, and in your NCERT biomolecule, they have mainly given you this beta wala part. Okay. This beta structure is there. I'll show you the diagram now. So this is going to be your beta ribose. Okay. So ribose is a pentose C5H10O5. C5H10O5. What's that? C5H10O5. Done. So bacha, this one more structure I would like to discuss here. This is deoxyribose. This is deoxyribose. So it is what? It is deoxyribose. It is deoxyribose. So what is the difference in deoxyribose and ribose? Look at this. So in deoxyribose, at second carbon beta, at second carbon, O is absent. Right, bacha? So O is absent. So here the formula is C5H10O4. So it doesn't obey the general formula. It doesn't obey the general formula. Am I clear? Am I clear? So this is your deoxyribose. Deoxyribose. Is that clear? What's that? This is your deoxyribose. Now you have to tell me option A, option B. What is it? Is it alpha deoxyribose or is it beta deoxyribose? That's what you have to answer. I will start the poll for 45 seconds. What's that? Is it alpha or is it beta? Quick. Is it alpha or is it beta? Quick. What's that, bacha? Is it alpha or is it beta? Speed up. Tell me quickly. Speed up. What's that? Is it alpha or is it beta? In the poll, answer it. Answer it in the poll. Quick. Yes, please. Yes, please. Quick. Sure. So it is simple. It is simple. It is alpha. It is alpha. Right, beta? So it is alpha. So that's how you need to understand that. Okay. So next thing that we have to understand is epimers. So before writing the definition, I would like to show you the structure first. जैसे ये देखो, C double bond O H. Look at the epimers here. Beta, what structure? This open chain structure is of? This open chain structure is of glucose. 1, 2, 3, 4, 5, 6. Now, here, वैसे तो I can write like this also, but चलो. See this. So this one is mannose. and this is the galactose. This one is galactose. Okay, ruk jao. So look at the structure. Look at the structure, everyone. So see this part. Yes, please. So if you compare them, what is the difference that you people can see? What is the difference that you all can see? So epimers, can I not say that epimers are the stereoisomers also? They are also the stereoisomers. Yes, please. Can I say that? Yes, Navya. They are also the stereoisomers that differ from other sugar, that differ from other sugar at only at one chiral carbon. ये देखो, rest everything is same. So they are differ from other sugar, right? हाँ, so other sugar in configuration only at, मतलब OH, H position is changed. So in configuration only at one chiral carbon. So look at the glucose structure and look at the galactose structure, right? So it will be, there will be the difference in the configuration only at one chiral carbon, bacha, while configuration at other chiral centers remain same. Rest everything is same. Only at one chiral carbon it will change, while configuration at other chiral carbons remain same. Okay, so that is what you need to know here. That is what you have to understand. So now one more question that you need to answer. Just 1 minute. Okay, so these are the epimers. This galactose, mannose, they are the epimers of glucose. So now answer this. What is this? D is fructose. Now you please tell me what this structure is. I'll give you 45 seconds. So if you are directly joining from the YT, right, then you can download the pylons and then you can answer this particular question. So quick, bacha. What's that? Quick. Yup, go for it. Go for it. So which structure it is? Sure, done. Bacha, it's simple. Yes, it is galactose. It is galactose. So 50% students, they are able to give the correct answer. Beta, see, carbon 1, 2, 3, 4, 5 or 6. So in the galactose at fourth carbon, that OHH position was changed. No. So if I have to draw the glucose here, that's going to be your glucose, right? That's going to be your glucose. Have a look. So please do not repeat this mistake. Pay attention. Do revise it. Very good, Pelaru. Very good, Sohani. Okay, so that's what you need to do. So up to this part, all clear? Chali. So now, beta, before starting the bonding part, here you have the structures and all. So only few terms I will discuss. Just now I told you, optically active. What does it mean? Optically active. That's what we have to discuss. Number one. Number two, whenever just say here, capital D is written. Bacha, in the chemistry, you will understand that in a better way. Capital D and small d, they have different meanings. They have different meanings. Right? They have different meanings. Just say capital D and capital L. Okay, capital D and capital L, they tell us about the change in the configuration of this HOH. They are change in the configuration, arrangement of these, right? Arrangement of this HOH. When you are writing small d and small l, right, which will be denoted by plus, small d by plus, small l by minus. So when it is small d, it is telling me about dextrorotatory. Levorotatory, L for levo, L for left. So if let's suppose a compound, right? If let's suppose you are having a compound, just say you are having plain polarized light. How can I explain it? Maybe you are having an idea of that too, right? You study that in the physics also. Just say in the plain polarized light, you will see that one component only. Just say light is going here. Just say light is going here, 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 here. But if it is having only one component in any direction, if it is having only one component, that is the plain polarized light. So whenever you talk about any compound, if it can rotate that plain polarized light, if it is rotating it towards right side, it will be dextrorotatory, right, beta? If it is the rotating it on the left side, then it will be the levorotatory, right? So these are the word that we have to discuss here. So plain polarized light, right? Plain polarized light, matlab light where the electric field or that vibrations are restricted to a particular single plane only, right? In that single plane only, that vibrations are restricted. Is that clear? Plain polarized light. You might have studied that in the physics too. Yes, beta. So where you will see that vibrations are restricted to only in a single plane, right, bacha? Single plane, it will not go here and there in this direction. Just say, let's suppose if there is a light, it's not like that there. It will not go. It will be restricted in one plane only, right? Just say there are substances from where when you pass the light, it becomes plain polarized. So all other components will not be there. It will vibrate only in one component. So maybe you have read that in the physics. In the batch itself, when we were having the classes, in that too, I have shown them the video also, right? So if that light is getting rotated, right, towards the right side, then it will be dextrorotatory. If it is the left side, then it is going to be bacha, it is going to be levorotatory. Is that clear? Is that clear? Yes, please. Done? Sure? So optically active, clear? If it can rotate the plane of optically active monosaccharides, optically active compounds are what? If they can rotate the plane of, they can rotate the plane of plain polarized light. Done, bicha? So plain polarized light, clear? So that will vibrate here, restricted to the one plane or one direction only. Okay, beta? Done. So if they can rotate the plane of polarized light, yeah, plane. Done, bacha? So if it will rotate clockwise, that is, so if it rotates clockwise, that will be bacha dextrorotatory. That is right side. है ना? Dextrorotatory. Plus, if anti-clockwise, then it will be left side, and then it is what? It will be levorotatory. That is minus. Is that clear? That is minus. Is that clear? Sure, beta? Done? Sure? Yes, please. Is it clear? Sure, everyone? Pakka? Done. So whenever you talk about the capital D or capital L, so it talks about configuration, right? Here you are talking about configuration. So if it is small d and small l, you are talking about optical activity. Okay, so never ever use capital D, small d interchangeably, right? They have different meanings. Is that clear? So you compare the configuration as per glyceraldehyde. Okay. Okay. So if OH is on right, capital D, OH is on left, at chiral carbon, it will be L. So that's it. This is enough for you. Now, next part is the reducing sugar. So any doubt? Any further doubt here? Yes, students. Any further doubt here? Pakka? Sure? Anybody having any doubt? So this is what you have to remember. So here you see OH position at chiral carbon. So OH right pe hai, so that will be capital D, and in chemistry you will read that properly, right? So here small d you can also denote it with plus, small l with minus. So it is for the right side, that is clockwise, and it is for the left side. That's it. That's it. Okay, so beta, now we are talking about the reducing sugars. I told you initially that let me finish these details and then we will talk about that. Reducing sugars. जैसे all monosaccharide या most of the monosaccharides are these reducing sugar. So what is the meaning of reducing sugar? Beta, they are having the free aldehyde or ketone group at, they have, right? They have free aldehyde or free ketone group. Do you know that? Yes. Yes, please. Do you know that? Do you know that? So reducing sugars, so this is the word that we have used. So they have free aldehyde or ketone groups. They have free aldehyde or ketone groups. And sometimes we also write it like this, that they have free anomeric carbon. So they have free aldehyde or ketone groups, right beta? So what are these reducing sugars? They have free aldehyde or ketone groups, bacha. Yes, please. Right? So they have free aldehyde or ketone groups. Yes or no? Which can reduce the mild oxidizing agent. Which can reduce mild oxidizing agent. So they can reduce these oxidizing agents, such as, in chemistry also you study that, such as Fehling solution, right beta, Benedict solution. So that is what we discuss. So in the chemistry also you study this particular part. Okay. Okay. Done. So that free anomeric carbon beta, that will allow the sugar ring to open in the linear form. That free groups will be there that can reduce the particular agent. Right? Right? Sure? So they can reduce oxidizing agent. So we can use it in the lab test. So many other tests are also there. Okay. So I will give you one example. That is what we have discussed in the class also. Just glucose. If glucose is present in urine, You know that that's a symbol of diabetes mellitus. This is known as glycosuria. So how can we test that? No doubt in the modern testing, we do not go for this only. We have other ways to figure it out. But what can we do? Just say, let's suppose here you have the urine sample. You are not going to taste the urine, na, ki urine is sweet or not. Hi, it is sweet, so glucose is present. We have to do some testing. So what will be there? Just say, let's suppose this is the urine. You want to confirm whether glucose is present or not. So you can put that particular, these mild oxidizing agents, right? You can put these mild oxidizing agents there. Are you getting my point? Are you getting my point? Just say, let's suppose if you will do the test with this copper based Benedict solution, right? That copper based test, just like Benedict solution, if you will put there, what will happen? The change in the coloration will indicate whether this glucose is present or not. That's how we can confirm that. So in chemistry, right beta, in chemistry lab, in that chemistry practical part, right, you will discuss that. So it is basically these free groups, they are capable of reducing these mild oxidizing agent. Okay, done. So you can read more about that. Done. So these reducing sugar can reduce CO2 positive too, right? These reducing sugar can reduce CO2 positive to C plus, right? So that's how they'll give us the results. We can detect that. So monosaccharides are basically the reducing sugars. Now the next part that we have is oligosaccharides. Next part that we have is oligosaccharides. So I told you they are on the basis of number of saccharides. That can be from 2 to 7. That can be from 2 to 7, isn't it? So I will give you the example of disaccharides here. So the very common examples that you need to remember is maltose, lactose, which is also the milk sugar, right beta, and then here you have example of sucrose. So if I talk about maltose, it will be glucose plus glucose. When two glucose they join to each other, that will be maltose. When beta there is the joining of galactose plus glucose, that is lactose, it is also the milk sugar. And when there is the glucose plus fructose, right beta, when there is the glucose plus fructose, then it is the sucrose. So maltose and lactose, they both are reducing sugars. And the sucrose is what? Sucrose is non-reducing. Sucrose is a non-reducing sugar. So sucrose is also known as the invert sugar. It is the cane sugar beta. It can, when it is in sucrose form, right, the rotation of plane polarized light is in a different direction. When you hydrolyze it, you will see that there will be a change in that in the plane of that plane polarized light. There is a reason behind. So we will discuss that. So these are the disaccharides. Now, how can we join them? I will just teach you the glycosidic bond, then we will end the class because I know you need the revision as well. So how can we join them, right? How will they join to each other? Beta, in the, right, so sugars they join. Yeah, these saccharides, they join by glycosidic bond. How do they join beta? They join by glycosidic bond. Which bond is going to join them? It is going to be your glycosidic bond. So how that glycosidic bond will form? We'll take a very simple example, right? Draw the glucose here. You will take the example of maltose first. So you will take the example of 2 glucose here. How many glucose here? 2. जैसे देखो बेटा, HOH, HOH, OHH, HOH, HCH2OH. So that's 1, that's 2, that's 3, that's 4, that's 5, that's 6. Then again, one, 2, 3, 4, 5 or 6. These are the glucose. So this first carbon of first glucose and the fourth carbon of next glucose. Beta, here what will happen? There is the release of water. What will happen? Ye dekho. OH here, H here. So what is going to happen here? The water will release, right? The dehydration will occur. It will be minus H2O, right? The water will release here. What will release, bacha? The water will release here. So what will be the resultant then? What will be the resultant then? So that's how the bonding will occur. Let me write it. Okay, so this is the glycosidic bond, right? This is the glycosidic bond which you need to see here, which you need to understand. Here you people can see. Yes, please. Ye dekho. So that's how these two glucose are going to join with each other. So this is your maltose, malt sugar found in the germinating seeds also. Ye dekho, that's how it will form. So can I not say that carbon number 1 of first glucose, fourth carbon of next, and both glucose here are the alpha. So this is 1,4 glycosidic bond, and you can also write it alpha 1,4 glycosidic bond. So it is the glycosidic bond which is formed by dehydration. It is the glycosidic bond, beta, which is formed by dehydration, and here this is how it is going to help that, right? This is how it will help to join these two, right? Join these two. Is that clear? Is that clear? So what do we have here? We have maltose here. So this maltose is nothing. It is the malt sugar, right, beta? It is even found in some germinating seeds. Okay, so it is two glucose molecules that will join. So alpha 1,4 glycosidic linkage is there. Then, bacha, you have lactose, which is the milk sugar, and it is in between galactose and glucose. Right, this particular, right, the lactose, right? So lactose will have. So lactose will have what? Lactose will have what? It will be having, right, beta 1,4 glycosidic linkage between the glucose and the galactose. So that's your homework. And one more homework that I'm going to give you is why sucrose is called as invert sugar. So it is related to chemistry, but I think you can figure it out. You are so smart, right? You can figure it out. So do check the bonding part there. Do revise it, beta. So this is all about today's class, right? So polysaccharides we will discuss later, because first you have to understand this information. So make sure you revise it. Now, if anybody is having any problem that why, extra content, anomeric carbon, chiral carbon, because this is the terminology we are going to use in this chapter. So if you are not aware of it, how are you going to understand that? Okay. So that's it, beta. This is all about today's class. Rest all we will discuss in the next class. So till then, take care. Bye-bye. I hope you guys have enjoyed the lecture. So if you want to be the part of our batches, here in the description box, right, there is a link. You can click on that. You can be the part of our batches. Arjuna batch is going amazing. Yaqeen NEET English batch is going amazingly. And students are too happy, right? So when you join us on the platform, you will get the chance to have infinite practice. Proper DPPs are there. Proper practice questions will be there, right? You will get the handwritten notes. Moreover, along with the handwritten notes, separate PYQ practice sessions are there. So there is one more session that I will be taking on this Friday, right, at 9:30 p.m. where we are going to discuss living world, biological classification, plant kingdom, cell cycle and cell division. Okay. So see you all then. Till then, take care. Bye-bye. Thank you so much, people. See ya. Bye-bye.